Today we will discuss about why the chemoresistance doesn't work or has very low efficiency in the pancreatic cancer or pancreatic adenocarcinoma, and how this gemcitabine helps to overcome chemoresistance to some extent. So first we will understand how this chemoresistance uh, has not been able to provide effective results on pancreatic cancer. Before going directly into this topic, let us understand or quickly revise few topics such as this EMT. So EMT is this epithelial to mesenchymal transition. So what happens in this transition is that cells that does not have the migration properties or metastasis properties will acquire the migration or metastasis properties. We can see that in the epithelial cells they are looks like a stagnant position, whereas in the mesenchymal cells they are stretched out as they are trying to move from one part or one place to another place. So this thing occurs when there is a reprogramming occurs within the cells that leads to the upregulation or epigenetic changes in a way that will promote mesenchymal cells. And next, we need to understand these cancer stem cells. The cancer stem cells are exposed low to the ROS. We can see that they are surrounded by the other cancer cells. So there is very low chances that the chemoresistance drug will reach to these cancer stem cells. And when the cancer stem cells are still uh, exist within the patients, there is always a chance of cancer regenerations in the later stage of the life in the patients. So this is some of the points that we need to understand this topic. So let us understand how this chemoresistance works. So when the chemoresistance drugs are usually supplied through the blood vessels that can be seen here, and this with the help of some carrier enzymes, these drugs will then move to the site of tumors, and when it's reached to the site of tumors, it will trigger apoptosis or other pathways that will reduce the tumor growth, or at the same time that will activate the death of the tumors either by triggering apoptosis or by reducing the food supply. It could be any one reasons. In the case of pancreatic cancer cells, what happens is that the enzyme, the carriers that helps in the transportation of the transportation of this chemoresistance drug to the cancer site is been down regulated. So because of that, the influx of the drugs to the site of these cancers is been reduced, and the drugs concentrations. So definitely the drugs concentrations are reduced that are able to reach at their target site. And since the enzyme concentration is low, the influx will be low. However, the efflux will be high, as the enzymes, instead of going to the target sites, they will they will form a bypass, and from the influx they will directly go to the efflux. So this is the one of the major reasons why chemoresistance are not that much effective. And the another reasons that I have showed that we have discussed initially is that the cancer stem cells are very less prone to be affected by the chemoresistance drug. So, however, some of the cells are able to reach to the cancer cells, and some of the drugs are able to reach to the cancer cells, and how they perform their functions is been described in is been described by one of the examples or by one of the drugs in the figure on the right hand side. So here the functioning of this gemcitabine is been described. So those drugs that are able to reach to the cancer cells, they entered into the cancer cells through this pore, that is HENT or HCTN. And once it reached to inside the cytoplasm of the tumor cells, they will phosphorylate through different pathways such as this uh, DCK, MPK, NDK. After that, it will get phosphorylated. This DFDC get phosphorylated by going through this pathways. And once these phosphorylates are this gemcitabine drugs are phosphorylated, it will reduce the DNA synthesis. So DNA synthesis will be reduced so that tumor cells will not be able to reproduce or replicate. However, there is a problem is that the DCTP, it will try to inhibit this phosphorylated DFDC. So it's a competitive reactions. And in order to reduce these competitive reactions, this function of this DFD CDP is also to reduce its precursor. DCTP's precursor that can be seen here. So in this way, the gemcitabine will act to reduce the DNA synthesis. So here also we can see that some of this DFD DCP has been utilized to reduce the competitive inhibition. So this will also reduce the efficiency of the drugs. So there are uh, two areas by which drugs efficiency are reduced in this gemcitabine effect. First, the amount of drugs that reach to the target site is reduced, and also the efficiency of the drugs has been uh, decreased by efficiency of the drug has been decreased by utilizing some of those drugs to reduce the competitive inhibitions. So this is how gemcitabine works to overcome the chemoresistance within the pancreatic cancer cells. So I hope you got a brief overview how this chemoresistance is not too much effective in the pancreatic cancer cells, and at the same time uh, the functioning of this uh, gemcitabine when it reaches at the site of this tumor infection. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.